Hello, friends. With me today in the dialogue is Dr. Abel Lawrence. He is Professor of Immunology and Rheumatology at Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. Dr. Lawrence, welcome.
Welcome.
Dr. Lawrence,
So,
why is it so difficult to get a vaccine against coronavirus?
vaccines development requires certain steps. First of all, there has to be a vaccine, and we need to know that the vaccine is effective, that it does what it is supposed to do, that it does not do any harm, and it does. And especially sometimes vaccines and immune response can be harmful to the host also. Moreover, this particular virus is a little tricky virus. It may not be very easy to develop vaccine, although we may be successful in making vaccine because the immunology has advanced quite far and we know a lot more about vaccines now. If you look at vaccines, how does vaccines work? Vaccines can work at two levels. It can produce antibodies and it can also create a very good cell mediated response which can then kill infected cells and therefore clear the virus. And in the case of antibodies, antibodies can also work at several levels. For example, in certain bacteria like pertussis and tetanus, etc., when there is a toxin, an antibody to the toxin can block it. But in this case of virus, there is no toxin to be neutralized. So what can an antibody do to a virus? An antibody can coat the virus and prevent it from infecting the cell. That is the first step. If it prevents a cell from getting infected, if you can prevent the virus from entering the cell by blocking its receptor, then obviously the virus infection will be reduced. But then sometimes it has been noticed that even when antibody coats the virus can still enter the cell. There are several ways it can do it. Then, and it has been shown that even when that happens, in case of many viruses, virus is still unable to infect the cell even after the antibody has coated it. That is because inside the cell, there are ways in which the cells can recognize viruses coated by antibody. That is, there are proteins which recognize such things and then tag with another protein called ubiquitin. Ubiquitin is just something which, if ubiquitin tags certain cells, those proteins, viruses are marked for destruction. This is like you mark this is for the garbage bin. So there is something inside the cell called a proteasome which degrades the cell, and once proteasome degrades this virus, and these degraded products can then be presented by the cell to the immune system, and a cell-mediated immune response can be launched. But this particular virus has a protein called papain-like protease. This papain-like protease can remove this ubiquitin from virus and therefore save itself. So imagine you have a police which has arrested somebody. That is, the antibody has arrested it and is sending it to the jail. But on the way, the virus is intercepting it and releasing it. So this kind of capability exists for this virus. But how relevant it is, we really don't know. We will need more studies, and probably in the coming months, we will know more about it.
Is this behavior peculiar to this virus, or there are other viruses?
There is always all these microorganisms like viruses, bacteria are always in a kind of competition, like a war between the host, that is us, the organism they're infecting. Evolution has this thing going on. Our immune system develops some defenses and then they overcome it, then we overcome them further. So this is and similarly immune system has several steps, and once a cell is infected inside the cell can detect something, whether an infection is going on by detecting things like this viral RNA, viral DNA, various things are detected, and it wants, one of the warning system is this interferon. So interferon will activate more of the things, and then once an immune system is activated, this cell mediated, the leucos, the WB, the lymphocytes will come and attack the cells and destroy this. And among the lymphocytes, the most important cells which are required for a vaccine protective immunity is something called CD8 cytotoxic T cell. There are other kinds of cells also, but if CD8 cells can actually efficiently attack a thing, then we can eliminate it very efficiently. And then once the cells which are infected virus comes down, the virus gets cleared. So if you cannot clear, then you can be chronically infected and chronically shed viruses. So in certain situations like influenza, the super spreaders who spread the virus for a long time are people who have defective cell-mediated clearance of the infection. So the clearance of infection is very important. So you have basically three steps in which virus can be stopped by a vaccine. And obviously the best one is if you prevent it from entering the cell. And then in many viruses, probably not in this one, you have another step even after it enters the cell. And so the antibody is the most efficient way to prevent, completely prevent, but that may not work in this case, or it may work. Time will know. But if it has already infected, the cell-mediated immunity can help. But the thing is, cell-mediated immunity cannot completely prevent infection. For example, if you have a some people have defects in the antibody immune response. Such people can get reinfected with viruses like chickenpox, which you should not get it again. But once but they will clear the infection because cell-mediated immunity can control it. So you can understand the difference. For a completely preventing, an antibody can help. So in this case, the problem is that sometimes in certain types of viruses where antibody is not fully protective, once the antibody coats the virus and enters the cell and the virus is unable, this actually can enhance the infection by allowing this virus to infect cells which may not be normally infected. An example is a dengue. If you are getting dengue for the first time, you get some viral fever and you get better. Suppose if you get dengue with the related strain of virus, it is not protective. What it happens is that normally some of the cells of the immune system which are not infected normally by the virus, they get infected because they actually swallow these viruses, which are which it thinks it can destroy, but it cannot. In fact, it gets infected. So this is called antibody dependent enhancement. So antibodies which are formed instead of controlling the virus actually make it more expanded, more overwhelming. And this is why if you take dengue, in the first week, first 5 to 7 days, patient is okay. Then around 5th to approximately 1 week, when the antibodies are coming into the circulation, person suddenly gets sick, and then within that 24 hours is the time when people with dengue can actually die. You might have noticed that. That is actually a classic example of antibody dependent enhancement. So when we make a vaccine, we have to make sure that antibody dependent enhancement doesn't occur. So to make sure of that, we need to know the antibody is targeting the right part of the virus which blocks its entry. So this is a challenge. Or if you can even a vaccine which can produce a good cell-mediated immunity. So as far as producing antibody is concerned, there are many ways. Even proteins, killed vaccines, proteins, spike proteins, fragments, so all of these can elicit immunity. The point is that to develop a cell-mediated immunity, the virus proteins have to be synthesized inside the cell. So that is why live attenuated viral vaccine, more sophisticated vaccine, even DNA vaccine and virus-like particles, they are better able to produce a cell-mediated immunity, which is like an insurance. Even if the vaccine doesn't stop the virus from entering, you still need a better immunity to. So it has been shown that in several animals like certain monkeys, some of the related Middle Eastern respiratory syndrome virus, SARS, they can actually, this kind of vaccine can produce amount of cell-mediated immunity and limit the multiplication, not completely stop.
WHO has talked about three kind of potential vaccines. I think they are for available for clinical trial now, and there are other 40 vaccines they have said that are in the stages of preclinical trial. How do you think this? I mean, do you think these vaccines will be potentially active or successful?
One of the remarkable thing is that the whole world has focused its ingenuity and resourcefulness in developing this virus. So the thing is, it's quite, but and different people are, following different strategies. Some of the strategies are relatively fast. So if you are using a particular protein, inactivated viruses, and maybe RNA, DNA, etc., relatively fast to make. The problem is that the faster ones may not, as I said, there are specific challenges to this virus. So, and so it may not necessarily be so if they don't work out, more vaccines will come. For example, I find some of the promising strategies which I don't think they'll be coming to, in the case, they'll be coming to trials that early in this case because they have to be created. For example, there are adenoviral vaccine. It's a model thing for which is adenovirus, something which is quite widely used for genetic engineering studies. You can, and then there is a measles vaccine based vaccine, etc., which have been tried in Middle Eastern respiratory syndrome, which is another coronavirus. So the biology of the thing may be different, but to make such which have shown immune response in animals, so they are actually, you can say, you can bet on those strategies because there is a prior case of successful vaccine development. Not they are not reached humans, only in animals. Then another successful strategy in the cases that in the case of Middle Eastern respiratory syndrome, they found that they had this antibody dependent enhancement when they used the spike protein. So to overcome this antibody dependent enhancement, they wanted to focus the immune response to those part of that protein which, if antibody binds, can prevent the virus infecting the cell. So that is by chopping off unwanted things and using only the part of the virus particle which is actually binds to the receptor. So it's called, that region is called receptor binding domain. So the receptor binding domain specific vaccines have been developed, which is basically part of the thing, and they have shown ability to block viral entry. But these things, as you said, is much more difficult than just taking something and injecting something. So actually, since we can't try one by one, all of these things should be tried in parallel. Which one were successful has to be mass produced and initially rolled out to high risk individuals, like those in the medical profession, police forces, people working in airlines, transport industry, essential services. Then then to the population, people maybe elderly. So that should be the strategy. So the world global strategy is actually a good one. Let's hope. But the thing is that you have to prove efficacy. You need to prove that they don't do harm. So
So
therefore,
will it take a year or a year and a half?
the estimate is that it may take 1.5 years.
Mm-hmm. Okay.
So that is why the slowing down of the spread of the infection is very important. We need to buy time.
Mm-hmm.
We need to buy time for development of a vaccine. We also need to buy time for manufacturing sufficient amounts of drugs. For example, there is a very promising drug called remdesivir. There is some trial that has preliminary data has come. The problem is that there is stock of only 5,000 medicine for only 5,000 people. The company can make for another 5,000 people using ingredients which are available. But how many people are infected?
We already crossed around
Even
9,000.
UK has some 80,000 people. So this is not going to reach anywhere. And to produce something like this in such large quantities, they say it takes at least 6 months.
Mm-hmm.
So if we can, so what is better, getting this infection now, getting it after 6 months, maybe when hopefully remdesivir and newer drugs may become available.
Mm-hmm. An international research report is doing round, which says that the countries which have BCG vaccination compulsory are least affected with coronavirus. So I would just like to know whether there is any similarity between coronavirus and the tuberculosis bacteria for which BCG vaccine is being used.
As you said, BCG is a bacteria, coronavirus is a virus, there is no similarity. The BCG vaccine, that is the tuberculosis heat kill bacteria, is used as an adjuvant to boost immune system during the time of vaccination. So, but they're not used in humans. We are immunizing an animal, they use. So people might be confused. That is because in terms of immunity is very specific. If you have if you get infected with one particular, such as say dengue, you have immunity against only that strain of dengue virus. You don't even have immunity against a related another strain of dengue virus. As I said, if you get another related dengue virus, you actually have a worse disease than the first time. So the immunity is not very generic. You have to have experience with that particular strain, either through an infection, which may be mild or severe, or through a vaccine. So nobody in this world except people who have had the disease can be immune.
Mhm.
So the very reason that we are having a pandemic is that human population has no immunity to this virus. Since we have no immunity, everybody is susceptible. That is the rule. That is why this is a pandemic. So the idea of immunity is a preposterous one, and it's a dangerous one.
Mhm.
It is dangerous because if you think you are immune, then you will not take things which we know can protect you. So it is not necessary that if you don't have immunity, you have to get it. If you don't have contact with an infected person, if you don't have contact with potentially infectious materials like if somebody has touched the fluids, aerosols, etc., you will not get it. And the most number one proven method of stopping an epidemic is the people who are infected should be isolated or quarantined. Isolation is a term used when you prevent you know somebody has a disease and you prevent that from contacting. Quarantine is for suspected persons. So if you can isolate everybody with coronavirus until they get cleared of the virus, then this epidemic will end then and there. So why can't we stop it? Because we are unable to trace and find everybody. Partly because in many large countries, the numbers are completely out of manageable size. That is why recognition of this pandemic potential early and attempting to control this before it went out of hand was the most important. Problem is human beings do not know how to predict the future. We always look at the past. We look at how many is there, not how much it can be. So we need, and secondly, so this is identification. So the testing, tracing is important, is part of identification and isolation of those individuals. Next, measures like social distancing, wearing masks, washing your hands, all these measures are proven to be protective. So we need to concentrate on that. Thinking of the immunity, boosting immunity while continuing to expose is actually risky. And if you look at various outbreaks, these outbreaks are occurred because people foolishly, for one reason or other, think they are immune. Maybe because they believe too much, whatever. So the thing is, these talk of immunity to our this thing, that is actually dangerous thing, because people may indulge in risky behavior which will be detrimental not only to themselves. The key thing to remember is, if you are getting it, you will be exposing your family members to it. You may have family members who are old, who may have hypertension. Maybe you may give to a family member or a friend who may then give to another person. So even if you may have a low risk, you may be young, your probability of something happening to me may be 0.5% or 0.3%. But you can give to somebody who is at risk. And then if you want this to end, then we want 0 people to have it. And this virus is going from humans to human. It's like a chain. We need to break the chain of transmission and end it.
We have been receiving a lot of tips to boost the immunity on social media, a lot of food stuff, even hot water. I mean, people are drinking hot water, they are not using air conditioners. Um, because you are an immunity expert, so just let us know, how can we build our immunity against coronavirus?
One is nobody has an immunity against coronavirus. So you cannot boost something which is not there. So let us be clear about that. So nobody can have immunity. But on the other hand, besides this, what is this adaptive immunity, which is specific to the virus, we have something called innate immunity. But those things, if you can correct something if it is deficient. If you have, there are various ways in which your immunity can be weak. For example, if you have vitamin D deficiency, you may have zinc deficiency, some nutritional deficiency, you have some disease like diabetes, all these situations weaken your immunity. So these are things to some extent you can do. You can vitamin D, you can take vitamin D, but then this only work up to correcting it. If you are not deficient, taking more of it is not going to boost it. So if you are immune, so for example, vitamin D is necessary for immunity to TB. If you are deficient, the risk of TB may go up. But that is why in the olden days, before any treatments were available, people with TB were put in, something called solarium, where there's a lot of sunlight. They didn't know about vitamin D, and that sunlight is actually producing vitamin D, but that was how it probably worked. So you can only correct what is deficient. So most people, if they don't have any, deficiency, you cannot boost your immunity and get out of this.
Having hot water or not using air conditioner help?
No. So if it has to be hot enough to protect you, then it should be hot enough to kill us also. So virus is living in so people with what is normal temperature with a fever, see this temperature of either hot water or the air without air conditioning is not hotter than what is our own body temperature. So how can, if we can survive and thrive inside our body, how can this kill the virus?
Mhm. Thank you, Dr. Lawrence, for talking with us. Thank you.